I'm Farida Koch and
I'm Sardava.
we are from the Shambi International School from 10th grade. And the name of our project is Treatment of Metals in Compared Analysis using Microbial Fuel Cells.
Polluted areas are tremendously increasing because of the impact of factories, poor recycling system, and especially human-based problems causing the irreversible problems in the nature. Today, the conventional method to recover the metals still require a lot of electrical energy.
In order to treat heavy metals, different methods were carried in this study. The following steps were conducted. Firstly, determination of polluted levels of heavy metals, which was done by our previous group. Secondly, constructing a microbial fuel cell. Thirdly, adding different metals to determine the voltage currency. And lastly, changing the parameters in order to observe the performance of MFC, such as substrate type, substrate concentration, and microbes.
One of the effective methods that we worked on is to use microfungi and vegetables, such as mucrofungi, onions, lettuce, carrots. This gave the best tolerance and absorption in the capacity of the heavy metals. So microfungi clean the soil for 20% and vegetables for 25%. So we choose the Tajik aluminum factory and the area near it for studies. According to analysis, we found out that the concentration of the heavy metals is 80%. So in the final of our experiment, we decreased it to 25%.
Media acts as a source of food for growth of microorganisms. The media prepared in this study was the nutrient agar media. The following constituents were added to this media: sodium chloride, peptone, and brief extract and yeast extract.
The steps of the projects are: the first step is find the resistant fungi types. The second step, find the heavy metals tolerated plants. The third step is the calculation of copper and chromium in determining fungi and plants. The last step is to examine your solution in the real conditions.
A dual chamber of MFC is built by using containers. We put to one of the containers soil and to the other water. And for both anode and cathode, we used aluminium mesh, and we separated the the containers with aluminium bridge, with salt bridge, which was made by salt and salt solution and agar. And we counted which multimeter was used to measure the voltage across the resistor.
The pH of the soil should be 7, so the microbes can survive, and this is the purpose of the sodium phosphate. The trials. In the first trial, we should add 400 milliliter of phenol inside the anode chamber, and we should add the sodium phosphate. 15 milliliter of Pseudomonas aeruginosa is also added in the anode chamber. The cathode chamber is filled with distilled water, and the voltage is measured regularly. In the second trial, we add 15 milliliter of Shawanaella putrescens. In the third trial, we add the mixture of 15 milliliter of Pseudomonas aeruginosa and Shawanaella putrescens. In the fourth trial, we add 400 milliliter of, and in the fifth trial, we add wastewater. In the sixth trial, we add our product, the ashes of the copper and chromium. In conclusion, we want to say that the microfungi cleaned the soil for 20% and vegetables cleaned it for 35%. So totally we cleaned the soil for 55%. After harvesting the crops, we turn them into ashes of copper and chromium.
It shows that no bacteria, no phenol, and no wastewater produced the best voltage currency. But our product, which was the ashes of copper and chromium, produced the highest voltage, which was 134 milliwatts. Thank you.